All right, biology. We're going to talk about the cell cycle today. So the cell cycle, this controls replication of somatic cells. So somatic, this word somatic here, just means body cells. Right. So the most of the cells of your body are somatic cells. There are things called germ cells we'll talk about later. And germ cells make gametes, which are sperms and eggs. Okay, so, but right now we're talking about somatic cells, right? So somatic cells, they're distinct phases. So most of your cell life is an interphase. You can see that as the big blue area of that pie chart. So most of it's an interphase. An interphase is the time between, right? So this is the time between cytokinesis and mitosis. So if we look at it here, so interphase is between cytokinesis and mitosis. Remember that, right? So it contains G1, S, and G2. We're going to talk about all those in a minute, right? So all those are part of interphase. So there are different types of questions. The longest part of the cell cycle is technically interphase, right? There's the longest part of mitosis, or the M phase, but the longest part of the cell cycle is interphase. And then there's the M phase. So there's interphase, and then there's the M phase. Mitosis, or cell replication. And the M phase ends in cytokinesis. So cytokinesis is often viewed as part of the M phase, but it's not. So if we're going to our roots, cyto means cell, and kinesis means movement. Right, or the cell parts are moving away from each other. We'll talk about that on Friday. Okay, so we'll talk about interphase first. Right, so we're going to talk about interphase first. Right. So 3 stages. There's G1. So G1 is growth and preparation for DNA synthesis. So this is G1 here. Right, growth and preparation. Now within G1, it's very important, is G0. So most of your cells are here. Waiting for a signal. And those signals are usually growth factors. So most of your cells are in G0 waiting for a growth factor, right? So make sure you know that G1 is growth and preparation for DNA synthesis. That's the only purpose of G1, right? Is the cell can grow and prepare for DNA synthesis. Remember, if we were talking about a bacterial cell, there is no G0. They just grow, divide their DNA, and divide. They grow, divide their DNA, and split in 2, right? The S phase, right, is DNA replication. So make sure you know that the S phase is DNA replication. We'll do a whole chapter on DNA replication. So at this point, right, it's really important to know this. So this is really important to know. Once you replicate DNA, right, so you have 23 pairs of chromosomes. So you have 46 chromosomes, right? When you repair them, or when you replicate them, you have 92. Right? You can't live with 92. You have too much of everything, right? So once you get to the S phase, right, we have to complete. So I can stop at G0, but once I pass G0, I have to go to the end of the M phase, or I have to kill the cell, right? So apoptosis is cell death. We'll talk about that. If something goes wrong in the cell cycle, your cell will hold a little marker out, right, on one of its glycoproteins. On the outside of the cell, it says, I'm defective, come terminate me, right, come kill me, right? And we'll talk about sister chromatins in a little while. But at this point, so once you get to S, you've got to continue. So I can stop at G0, but I can't stop. And then G2 over here, right? So after we replicate DNA, then G2 is relatively short. It's just doing the final checks and preparation. It's still growing. Really what we do there is we grow and we check for DNA, okay? G2. So cell cycle control. Right, so checkpoints, the stop and go ahead signals received. So there are different stages. So there are 3 checkpoints. There's checkpoint G1, G2, and M, right? All involve DNA, right? So the reason why they do is because remember DNA controls protein synthesis, and proteins control what happens in the cell. And if DNA goes wrong, then cells go wrong, right? So if DNA goes wrong, there's a chance for cancer or something bad in that, right? So in G1, right, so the G1 checkpoints, it checks for size, nutrients, DNA damage, and growth factors. I put growth factors in another color, right? Because it'll check, in G1 it'll check for size, nutrients, and DNA damage. And if all those are good, it still waits. And it'll wait for growth factors. And growth factors are substances that it needs to build the chemicals inside the cell to get to the S, right? So if it doesn't have the growth factors, it stays at G0. Okay, so that's the important part. If the growth factors aren't there, and size is good, nutrients are good, DNA damage is good, then it'll stay there. Right? The S phase, right, is DNA replication. So during that, it's just going to copy the DNA. It's going to put it in different areas, right? And then it's going to usher in G2, the G2 checkpoint, right? Cell size and DNA. Right? It's going to check both of those. If both of those are good, it's going to continue. It's not going to wait for growth factors because growth factors are just to get past G0. In the M phase, it's going to check for spindle, right? Again, it's going to check for DNA. Make sure all the DNA is lined up and it's all going to be separated fine. Right? So if any of these checkpoints, right, so if any of these checkpoints don't match up, then the cell has one choice. Apoptosis. So if any of the checkpoints are incorrect, right, and can't be repaired, then the cell will signal apoptosis, which means death. So part of cancer is that this mechanism here doesn't work, right? We have errors in the apoptosis gene, which is the P53 gene, by the way, right, is not functioning, so it just continues. Okay. So the M phase. We'll talk about the M phase in great detail on Friday, but the M phase, or next time I see you, if it's not this year. So the M phase, 4 distinct, right? Remember, PMAT. All right, it's really PPMAT because there's a pro-metaphase, but for you this year, prophase, metaphase, anaphase, and telophase. Right? The purpose, right? The purpose of the M phase. So the purpose, I'm going to put that over here, is division of the nucleus. That's the purpose of the M phase, because we've replicated the DNA by the M phase. And the purpose of it is to get my chromosomes, right, and to separate them. Right? That's the purpose of the M phase, is to separate the newly divided chromosomes so I can get them into 2 cells. Right? And it forms 2, so these are chromatids. These right here are sister chromatids. Right? Because they're copied. And these right here are daughter cells. Which are clones. Like identical twins, same DNA. Right? So it's sisters and daughters. There's no sons and brothers. Okay? Sisters and daughters. Cell cycle control. So we talked a little bit about it. All right, 3 checkpoints. All right, so we already talked about those, so I'm not going to go back because you can go. Right? But remember, we have a checkpoint at G1, G2, and M. The only place without a checkpoint is DNA synthesis. The only thing it's doing there is DNA replication. Right? But they all check for DNA. Right? So all check for DNA. And if the DNA is correct, then it splits. If the DNA is not correct, then it can't be fixed. It'll signal apoptosis, which means cell death. So molecules associated. So here's the important part. So you have CDKs, cyclin, and MPF. All right. So I'm gonna try to make this easy. So cyclin-dependent kinase. ACE means it's an enzyme, right? So cyclin-dependent kinase. If you look at this picture on the right, so cyclin-dependent kinase is floating around in the cell cycle. Right? It's always present in the cell cycle. Right? Now, cyclin, right? Cyclin is a molecule, right, that's activated. Right? So if you look at here, right, cyclin is being built, right, at the start of G1, or at the end of M phase. Cyclin is being built at G1. So cyclin, let me get a different color here. Produce right, so it's produced at the beginning of G1, right? So if you see here on this chart, right, the beginning of G1 is right here, right? So cyclin beginning of G1, it starts to accumulate, right? So cyclin then will bind to CDK. So when growth factor is present, S cycle. So the S cycle happens. So if you look at here, so at the S cycle, my CDK, my cyclin is right there. Right? CDK still accumulates. Or cyclin. Right, so that says it accumulates here. So it accumulates. So at G2, oops, I lost my screen there. There we go. So at G2, right, we're right here at G2. Right, if you look at that picture on the right. Right? So when cyclin concentration is sufficient, so it binds a cyclin-dependent kinase to make MPF. And then MPF, right here, is mitose promoting factor. So this starts the M phase. And that's where the nucleus is going to divide. Okay, so that is right here. So it starts the M phase. And that MPF, the really important thing for the MPF, peaks during the M phase, initiates mitosis, aids in disassembly of the nuclear lamina. And that nuclear lamina is the nuclear membrane. So essentially what the MPF does is it takes apart the nucleus. Right? It allows the DNA to separate and move into two cells. And then it's switched off. Right? It's switched off during anaphase. Right? So it allows the new nuclear lamina to come together. Okay? It's a lot there in that. But to look at, so it's switched off here. So at the end of the M phase, cyclin is at its lowest point again. Right? And then it just starts to build. If you're stuck at G0, right, then we'll stop producing cyclin for a while. So molecules associated with the cell cycle, right, we have growth factors, right, so secreted by nearby cells, and they're called a mitogen. A mitogen causes something to happen. So in court, they can talk about mitigating circumstances. What were the things happening that made you do what you did? So a mitogen caused something to happen. Growth factors cause cells to replicate, right? So it's secreted by nearby cells. So if you get a cut, they secrete growth factors to make cells nearby grow and fill in where the cut is, right? So remember, they allow the cell to proceed past the G1 checkpoint. So these are, by the way, these are what they call, because you'll need to know this for your test, internal controls. So internal controls. So internal controls are checkpoints. So that's the G1, G2, and M, and growth factors. So those are our internal controls. So those are controls within the cell, right? External controls, right, outside the cell. Density-dependent inhibition, which means that cells stop growing when overcrowded. So if you look at this, if I have, look at picture number 1, I have 4 cells there, 5 cells. They will grow until they reach number 2, and they will stop growing. So once the borders touch each other, the cells no longer secrete growth factors. Right? They say, okay, we've got a layer. That's what we're supposed to have. We're not supposed to have a lump like a tumor. We're supposed to have a flat layer. If you scoop some of these cells out, they again will grow, right, until they form that flat layer. And the other is anchorage dependence, right? In order for cells to divide, they have to be attached to something, right? So if one cell breaks free, it'll just float, and it's not supposed to grow, right, because it doesn't have anchorage, right? So that's the problem with cancer. So cancer, these cells lack normal external controls, right? They will form a lump. They do not have density-dependent inhibition. So cancer cells lack. Right? So cancer's in stages. So this might be stage 1. Stage 1 and 2. Right? Later, I might get a cell that breaks free from this. Right? So right, stage 4. Right, stage 4 goes 1 through 4. So 3 is kind of a transition period of irregular borders. They say where it's starting to leave. But stage 4, right, cancer is free to move. Right? So this is called metastasis. Right? So cancer lacks both internal controls and external controls. So what we say here is okay. Well, that's it for today, I believe. Yeah, that's it for today. And I'll talk to you soon.